From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on April 25th, 2024. Hello, Micronauts, and welcome once again to today's Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today, believe it or not, is the 38th episode of Matters Microbial. Those of you who watch and/or listen and spread the good microbial world to others, many, many thanks. I recently showed a video of my bioluminescent firefly petunia plant on an earlier episode. This week, I took my plant into a darkroom and set up my fancy camera. As you can see, the flowers glow brightest, but the leaves are also all aglow. It's just amazing. Speaking of glowing things, I had to grow up a culture of Photobacterium leognothi, which my first-year students could paint with. Seen here, the flask gets much brighter when I swirl it. The luciferase reaction requires oxygen in plenty. When I have a glowing flask, I often ask if my first-year students are interested in a Lux selfie. That is, they hold the swirled flask near the face, and I take a long exposure. And here are 3 nice examples. This class really understood my different sense of humor, I think. I asked the students to paint with luminous bacteria a last time, sort of as a goodbye to me and the class. One student painted a glowing tardigrade, near and dear to my heart. One student wrote a thank you note to me in living light. A third student created a nice diagram of a cell. And finally, one student who was very fond of my Kenyan Sandboa laboratory mascot, Baby Carl, created a glowing snake. Yesterday was the last lecture of the very last time I will be teaching introductory cell and molecular biology here at the University of Puget Sound. In the fall, I start a 2-year phased retirement, teaching in one semester and focusing on research for the other. Yes, at half salary. I have many conflicting thoughts on my future, of course, but my wife, Jennifer Quinn, painted a lovely remembrance of my time teaching these courses at Puget Sound from 2005 until 2024. Most of you know how much I adore microbes, and I hope that I've made it clear that, unlike in the lab, most microbes live in association with other microbes. They interact in positive and negative ways. This concept, called socio-microbiology, is very interesting to me, and I'll put a link to a good review paper in the show notes. But how to study these associations? 1 at a time? 2 at a time? And this, in fact, takes us to one of my favorite foods, cheese. It turns out that cheese is a highly interactive community of microbes working together toward what is, to me, a tasty goal. Some scientists study and dissect the associations between different members of such a cheesy consortium. So, I'm extremely excited to introduce Dr. Rachel Dutton of the Astera Institute to the podcast to tell us about her journey in science, her work with cheese communities, and new directions in research. Welcome to our Quality Quorum today, Rachel.
Hi, Mark. Thanks so much for having me today.
Oh, it's been on my bucket list. I don't know if I like the word bucket list suddenly, but what I will say, it's been on my list to have you perhaps on the podcast because you know I adore your work. Incidentally, following some of the instructions you sent me a few years ago, my microbiology students were able to isolate some microbes from cheese and got the usual suspects.
Amazing.
But I will tell you, that smell? My students made me promise never to do it again because it was very strange. It smelled like the cheese, but there was an overtone of something that was not so nice with it.
Yeah, like feet, maybe.
Yes, in fact, like feet. That's pretty amazing.
That's how you know you're doing it right.
Well, and that is what I told them. And right now there are several, hopefully former students of mine watching and or listening, holding their noses in memory. And as you know, when a microbiologist says something is a little bit smelly, it means something.
Yes, exactly.
So, Rachel, I think that you have an unusual background. Would you mind telling us about the path that led you to, as we said, to cheese and Brie-ond?
Yes, I'm one of these people who, didn't grow up knowing they wanted to be a scientist. So neither of my parents went to college. I was a first generation college student. I, didn't know that doing science was something that I was even interested in, or that, was a thing you could be when you grew up, a scientist. So I went to college and I started out actually studying communications. And I think that's something that I've brought with me throughout my career, is this interest in communicating, what I'm doing to the public. But I sort of on accident took a microbiology course for non-majors. We had to, at a liberal arts college, I was at UC San Diego for undergrad, and, I had to sign up for some other elective. And the one that was available at the time was a non-majors course in microbiology. And it happened to be taught by, at the time, a new assistant professor, Kit Poliano. That was the first time I learned really about microbes as a freshman in college. And that class had made a really big impression on me. And, I stayed in my communications major for like another year. And then I started thinking, okay, actually, I don't know if communications is what I'm supposed to be doing with, with my life. And started thinking about that course that I had taken and thought, okay, well, is this the thing that I should be doing? Should I be, learning more about microbes and more about science? And so I went and, I emailed Dr. Poliano and asked her if I could volunteer in her lab. And I had never had any research experience. The only science class I had taken was her non-major class. And she, I guess somewhat surprisingly said yes, that I could volunteer. And that was really, I think, a life-changing moment for me, because I started volunteering in her lab and I had this feeling that, like, this is what I'm supposed to be doing. I felt like I was in my element for the first time. And I fell in love with microbiology, fell in love with microbes. I changed my major from communications to molecular biology, which is not something that most people do. And I think, apart from just learning about microbes and how fascinating and unexpected they are and how powerful they are, I think the other thing that really drew me into science was the fact that we were doing science as a team. And I really loved that sort of community and team element to, the work that you do, that I didn't feel like I was getting, that in other, majors and disciplines that I had been exploring. And so, that sort of just completely changed my life, having that early, very lucky experience of being able to work in a microbiology lab. So then I graduated, went on to do my PhD in the lab of a classic bacterial geneticist, John Beckwith. And so I was going down a path basically of studying model organisms. So in Kit's lab, we studied Bacillus subtilis. I had worked for 2 years at UCLA in Jim Gober's lab studying Caulobacter crescentus, worked in John Beckwith's lab studying E. coli. But at the time, when I started grad school, it was around the time when people were starting to do a lot more environmental sequencing. and really discovering the true diversity of microbes that are out in the world. And I was just absolutely fascinated that, okay, yes, we have a handful of species that we're working on in the lab, but there's all this like immense diversity of microbes, and what are they doing out there? So I got more and more fascinated with microbial diversity and starting to think about, okay, how do I play a role in understanding what's going on out there in the world of microbes? And so, again, I was really fortunate as a grad student to be able to go and take the microbial diversity course at Woods Hole, which was another sort of game-changing experience for me. Got my first experiences working with microbes out in the salt marshes, and bringing them back into the lab and trying to study them. And that really sort of set the stage, I think, for me to start thinking about other environments and how I might be able to work with them. So I don't know if you want me to stop there or keep going.
No, that's wonderful to tell people. A couple of things to emphasize. The first is some folks have an unusual idea of where scientists originate, and there isn't some factory outside Peoria where all scientists come from. It's a profession like any other. I too was a first generation person. And it's funny because if you come from that background, you don't know how you become a scientist of any kind, let alone a microbiologist.
Yeah.
And everything you're describing is an echo of things that lots of us, including me, have experienced in lots of different ways, though I didn't work with John Beckwith, let's be clear. So on this subject in general, as you're casting about for a system to study for yourself, that could meld these interests in, and I would say, model systems to actually understand what's going on one level. With community microbiology, you chose cheese.
Yes.
And why did that happen?
Right. So it's completely not obvious because I think, looking back, most people would have assumed, okay, you studied cheese, you must have been a food scientist or, I don't know, or at least a environmental microbiologist of some kind. But no, I was an E. coli geneticist. And exactly like you said, I was really searching for something that was at the intersection of experimental models where you could really take things apart in the lab and understand how they work, and something that would give us a glimpse into microbial diversity and what was happening. And so I had there's a number of things that came together that led me to cheese. I personally am obsessed with food. So was a sort of budding foodie when I was in grad school. I love to cook, love to eat, love to explore the world through food. And so was learning a lot about food. I was also really interested in science communication. So I was doing a lot in grad school on communicating science to the public, and started even using food as a way to educate people about science. It's just a really amazing platform to engage people. And so I had already started thinking about food just from a personal interest and interest in science communication. But the one thing that really, I think, was like the moment that pointed me towards cheese was I had come back from the microbial diversity course. And in the course, we had studied salt marsh mats, the microbial mats in the Sippawisset salt marshes down in Cape Cod. And I was fascinated by those mats. They're just beautiful. They have these beautiful layers, super complex communities. But they're in my mind, there was no way I was going to be able to study those. They're very complex. Anyway, so I came back, and we had in the Beckwith lab, we had a long history of people coming from France to study in the lab. And so we had a French postdoc who had just come back from a trip to France, and she had brought back a piece of this French mountain cheese called Tome de Savoie. And it's this beautiful rustic cheese aged in the mountains. And we were having a little wine and cheese tasting in the lab to celebrate her coming back and bringing us these treats. And we sliced into the tome de savoie. This was in the like lab kitchen area. And when we sliced into it, the rind had these layers, these like different colors of layers that looked like a microbial mat. And so I took a piece of the cheese in the rind and I ran back into the lab and I started culturing it. I made dilutions. I basically did what I learned how to do in the microbial diversity course, which was, take a sample from the environment, do cultivate different cultivation conditions, incubated the plates, and opened the fridge a couple of days later, and it smelled terrible, which is how I knew I was able to culture those things. And so it kind of, all these things came together and sort of presented itself to me as this opportunity to study a community that grows in a way that's, not that different than microbes in the environment, and is likely much simpler, is an environment where humans have been cultivating it for thousands of years. So I figured, well, we should be able to grow them. And then spend a couple of months just kind of playing around with the system while I was a grad student and formulating these ideas, and eventually wrote a proposal for an independent fellowship position. Got this position as a Bauer Fellow to start my own lab based on studying cheese as a model microbial community.
So to break in really quick, this is a really important thing for people to understand, is that if you wanted to study, say, the human microbiome, that's super complex. And I have such admiration for the people who do it. But you took the skill set that you learned not as an ecologist, perhaps, because ecology is always hyper complex, but instead something that was manipulable. That's what you were looking for.
Exactly.
And just as when people are studying biofilms, which we've talked about before on this podcast, they'll look at one organism. That's not how biofilms exist. So before you move on, I thought it would be good if you just spent a couple of moments telling the audience about microbes in cheese.
Yeah.
This subject came up when I was talking to the producer. So something that everyone, no matter what their level, is going to get, like Swiss cheese versus cheddar versus, of course, Roquefort.
Yes. So I can do a very condensed cheese 101. So cheese is a fermented food. Similar to other types of fermented foods, you might think of kimchi or sauerkraut or sourdough bread, beer, wine. They all rely on microbes for their formation. And so the fermentation of cheese starts out by fermenting milk. So you take milk. There are either naturally occurring bacteria, these are lactic acid bacteria, or the cheesemakers will add them. And those bacteria basically eat milk sugar, so lactose. So I like to think of them as like the lactose-tolerant microbes of the world. So they can break down lactose in the cheese, use it for energy, and convert it to acid, lactic acid. And that sours the milk, it decreases the pH, and that causes the milk to start to coagulate. Now, if you just have milk and lactic acid, you basically have yogurt. Lactic acid bacteria are what you find in yogurt. And that's sort of like basically converting milk from like a liquid to like a gel. But to make cheese, usually then you'll take it a step further and you can heat the milk or add an enzyme called rennet, which is a protease traditionally from the stomach of a calf. And that breaks down the protein further and allows it to coagulate even more. So that plus heat plus pressure, you're pressing out the liquid and you form a solid. So you take liquid milk to a solid state. And then you have fresh cheese. And now fresh cheese is delicious. It's tart. So something like a fresh goat cheese like Chev would be something like this. But then where it gets really fun is you can now take that fresh wheel of cheese and put it in a damp, dark cave and let microbes grow all over it. And that part of the cheese making process is what I have studied. And that is where you get the introduction of a ton of diversity of organisms and resulting in a ton of diversity in textures and flavors and smells. And so depending on the way the cheesemaker treats the cheese at that point, they can just sort of put it on a shelf and sort of let it be. They can wash it with a salt brine solution. They can, poke holes in it to let little bits of oxygen in. That changes the environment for the microbes. And really what cheesemakers have learned over thousands of years of cheese making is how to very precisely manipulate microbial communities to do what they want. So they can make very small changes in the pH and the temperature and the moisture. And that changes what types of microbes grow and how they grow. And so, you can have things like a blue cheese where there's Penicillium roqueforti inside, and it grows where there's just a little bit of oxygen. It's a microaerophile. There are the washed rind cheeses, which are washed in a brine solution. That promotes the growth of the really smelly organisms. And actually, they're similar to that salty, moist, acidic environment is similar to our skin. And so a lot of the body odor smells are similar to these types of cheese smells, because it's the same types of microbes that grow in those environments. And you can have very simple cheeses like a fluffy white brie or camembert, which are colonized by primarily Penicillium species. Those are kind of the main types. And then the fun one, Swiss cheese, is Propionibacterium, which grows inside the cheese. It's an anaerobe, so it doesn't like oxygen at all. It eats the leftover lactic acid from the lactic acid bacterial fermentation, and it produces CO2 gas as its byproduct. So basically, inside the cheese, everywhere one of these Propionibacterium little microcolonies is growing, they're producing gas around them, and those are the holes that you get inside the Swiss cheeses. So that's a quick primer.
No, and it's fascinating. I know that when I teach microbiology, one of the things that I point out is there are certain byproducts of metabolism that give rise to the taste of cheddar cheese. I've forgotten what the chemical's called. I'm sorry. but you know what I mean.
Yes,
and this is why it's such a fascinating subject. And I believe that I can put up in the show notes a nice primer to Cheese 101, as well as a couple of videos that you put together that I always show my students. So a couple of questions involving, I think, things that everyone hears about. And first one is cheese something that can generally be consumed by lactose intolerant people?
Yeah, that's a great question. I often get that, I can't eat cheese because I'm lactose intolerant. But actually, within the first couple of days of cheese aging, most of the lactose is used. I mean, lactose is a sugar. Microbes love sugar. So they are growing and eating as much of that sugar and breaking it down into more digestible components. Within the first day of making cheese, that's part of the fermentation process. So I would say that for people who are lactose intolerant, anything that's been aged, like a hard cheese, is not going to have any lactose in it.
That's really interesting.
Yeah, soft cheeses can still have small amounts of lactose because they have a lot of moisture, and that can the whey, the liquid in the from the milk, has more of the lactose in it. So the harder the cheese, the less lactose, or basically, there's undetectable amounts of lactose in any hard cheese.
Interesting. That's really useful for the listeners and viewers. Related question, and you've just touched on this in terms of lactose, glucose and galactose linked together. What other changes take place in what started out as milk? In other words, are there extra nutrients being added or taken away? Yeah,
so there's tons of different types of transformations that are happening of the milk itself. So like you said, the sugar, the lactose is getting broken down and digested and converted to acids. The proteins are getting broken down into smaller and smaller pieces. So you get the formation of peptides and then amino acids. Those can have different types of flavors and aromas. the fats in the milk are broken down into free fatty acids and then smaller volatile molecules that you can smell. So those are all being metabolized. In the process, the microbes are growing. They are creating their, because they are growing, they're creating vitamins and nutrients that they need to grow, and then they die and release those in the process. And so it's like any other fermented food where you get all of these benefits of the microbial growth and metabolism and all the nutrients that they're producing, as well as a complete transformation and sort of pre-digestion of the food itself to much more smaller, like more digestible components.
So generally speaking, the characteristics of cheese-making bacteria, I dislike that term. I should just say lactic acid bacteria. You've mentioned, of course, that they like a lot of sugars of one form or another, in this case, lactose. They have a uneven relationship with oxygen.
Yeah.
Some of them tolerate it. Some of it can't stand it.
Right.
Sounds like my social life in college. And are there other characteristics we should know about?
Yeah. So there are a couple different groups. So there's the lactic acid bacteria, which are doing that primary fermentation of eating the lactose and converting it to lactic acid. Those microbes are can be tolerant of oxygen, but don't love it. But then there's this whole other community of microbes that grows during the aging process, which is what my lab was focused on, the rind associated microbes. And those are all aerobes. So they like oxygen. They like growing on the surface of the cheese. And they have completely different metabolic properties. They are helping break down the proteins and the fats. They have all these interactions with each other and the fungi. So very different. If you think about like the tree of life and of bacteria, the bacterial portion of the tree of life, you actually, for relatively simple community, you have quite a bit of taxonomic diversity sampled across the bacterial tree from these different groups of organisms.
Can I ask a difficult question?
Yeah.
Is it bad that I like to eat the rind?
No, the rind is great. The rind, yeah, all cheese rinds are edible. I would say not all of them taste good. But something like a brie or camembert, I would definitely eat it. It has its own sort of mushroomy flavor. In some cheeses, the flavor gets more intense the closer you are to the rind because those microbes are producing a lot of flavor molecules and doing a lot of the metabolic activity of digesting the cheese. And so you can kind of like dissect a cheese and like eat it, closer and closer to the rind, and you can see the flavor changes as you get closer.
You are a gourmand.
Yeah.
We're talking about the human gut microbiome a lot. Is there any evidence that the cheese microbes, when we consume cheese, are communicating with the gut microbes, or do they not make it through the stomach?
Yeah. So I don't know that there's any evidence that we have that they're communicating with the gut microbes, but actually we did a study. This was published maybe 10 years ago now with Peter Turnbaugh's lab, as well as with Lawrence David, so both human gut microbiome researchers. And it was a human diet study where we wanted to know how quickly the human gut microbiome could change upon different diet types. One of them was a vegan diet, and the other was more of a carnivore diet where we had cheeses that I had selected for the diet study for human participants to eat. And we then took the fecal samples from the people who had eaten these different diets and tried to see if we could culture any of the cheese microbes from the fecal samples. And we could actually, several of the fungi and bacteria were able to re-isolate from the gut microbiomes, like fecal samples of the people who ate those cheeses. So they can survive that environment. I don't know if they're doing anything while they're there, and I don't think they're colonizing the gut microbiome in any way.
Just passing through, as it says.
Passing through, yeah.
Sorry about that. That's very interesting. And related to this, and I think you answered this question, but I want to address it. Am I correct in thinking that most of the bacteria that we believe are pathogens, they have trouble becoming established in cheese?
Yeah. So cheese is overall very safe. There are a lot of fermented foods. The reason that people have made them for centuries, for thousands of years, is because they create conditions that prevent the growth of most pathogens. So the acidification prevents the growth of many pathogens. The salt prevents the growth of many pathogens. So there are these sort of conditions that are prerequisites for the formation of these communities that actually prevent the growth of many pathogens. There are, however, some pathogens that can make their way into cheese. Listeria is one of the biggest problems. It likes to grow in the sort of cold environments that cheese is aged in. There can be Staph aureus can contaminate cheeses. So there are occasional food safety problems with cheese that cheesemakers have to be on sort of alert for and do a lot of food safety diligence to make sure that their products are safe.
And I know that many of the problems have been with softer cheeses.
Yeah.
And that makes me think about the level of acidification because of that. But regardless, I think it's important to have safe food.
Yes.
And most fermented foods have been around for thousands of years for that reason.
Yes, exactly.
It's so funny. I don't like to make fermented foods for myself, but I'll go buy fermented foods.
Uh-huh. Yeah. It's fun to make them. It's I have experimented at home with making a number of different types of fermented foods. It's, you're basically growing microbes at home and doing little mini experiments, and then you get to eat them. So,
but yeah. This is a great introduction to people. We have all kinds of listeners and viewers, people that start, and I call them new micronauts, as opposed to people that are more experienced micronauts. And what I like about this topic is there's something truly for everyone there.
Yeah.
And it's delicious.
Yeah, and I think that's one of the things that I love about cheese is that it's a microbial environment that you can interact with. You can see the microbes, which in most cases in the world, you have to use a microscope to see microbes. But on the surface of a piece of brie, there's a microbial community, and then you can smell it, you can touch it, and you can eat it. So it's a very interactive microbial community.
And then you went on, did you not go on to work with cheesemakers a little bit?
Yes, I spent a lot of time working with cheesemakers. I actually, before I started my lab, I spent 2 months working on the farm at Jasper Hill in northern Vermont. I made cheese, I worked in the caves, and yeah, basically have collaborated with them for 13 years. They're very, they make some of the best cheeses in the world. I mean, I'm biased, but they're award-winning cheeses, and they're wonderful people and very excited about science, and have been the source of a lot of the sort of environmental samples and the field work that we've done over the years is from their caves.
So let's be really clear on this. I'm just saying that it's Doc Martin approved. I have actually purchased some of their material, and it is indeed lovely.
Yes.
So I just wanted to say that. And then this is what's interesting, is that you really have bridged the gap. And I see some environmental microbiologists do this, but this is good. You've seen both sides of it for your model system. So the real question becomes, you can dissect out the community because unlike the situation, say, in the human gut, it's possible to grow, I wouldn't say all, but most of the members independently of one another. Would you agree with that?
Yes. Yeah. So, and I have a slide about this to show basically, what we can do with cheese, which is, I would say, fairly unique, and this is true for many fermented foods, is we can actually start with the environmental samples. Our field work is going to cheese caves, which is pretty luxurious field work. But and then we can bring them back in the lab and culture them. We can do that on fairly standard media. They like to grow in aerobic, rich media, and which is typically what we use as microbiologists as our default. And then so we can sort of create culture collections from cheeses. And one of the things that we've done from the very beginning, in part because my first postdoc was Ben Wolf, who is a mycologist by training, and now has his own lab at Tufts. But he really taught me and taught us how to work with fungi. And so a lot of the time we are isolating both the fungi and the bacteria from these communities, which in many cases in studies of microbiomes, people focus really solely on the bacteria. But cheese really offers us a nice opportunity to include fungi in our understanding of microbiomes. And so we can create these beautiful collections of different fungi, and you can see in the pictures these like beautiful fuzzy, colorful fungi as well as bacteria. And then we sort of wanted to know, could we put them back together and make communities in the lab? Because that's what we wanted to use this system for, to understand what are microbes doing when they're growing together compared to when we're studying them in isolation. And so we constructed these in vitro cheese communities. So we basically take fresh cheese from the caves, we freeze dry it, grind it up into a powder, mix it into agar media and autoclave it, so it's sterile cheese-based medium. And then we take from our culture collection these like beautiful individual isolates and we mix them together back into community-like mixtures and grow them, and can actually rebuild communities in the lab that mimic even ecological succession patterns that we see in the environment. So it's a really amazing system to be able to kind of dissect and reconstruct communities.
I want to break in and say that I like what you said about ecological succession, because this doesn't occur to many people until they think about food in general. So if you look, for example, of how sauerkraut is made, there are waves of different microbes where one group of microbes kind of prepares for the next one to come through, say by dropping the pH or say by producing a particular quantity. And so this is a wonderful test. And obviously, you cannot eat your lab cheeses.
No, unfortunately.
Good.
Or fortunately, I don't know.
Well, I don't know either. But the point is, you can still study how they're interacting together.
Yeah.
And to me, that's really interesting. I am cursed with very good smelling. What would I say? What would I call that? I'm like a bloodhound with this. So I'm really sensitive to bad smells. So for me, I'm wondering, could I detect the correct combinations, because there'll be echoes of what the final cheese would be? That's what I wonder.
Right.
I mean, now, true, I'm not a bloodhound, but that's such an interesting idea. And what I like about it is it's amenable to everyone, and it reflects things that we know about fermented foods in general.
Yes. Yeah, the succession is a really important part of cheese making. And one of the things that, still surprises me is how reproducible it all is for all of the fermented foods. I mean, in many cases, even in cheese making, a lot of the cheeses that we studied were made in very traditional artisanal ways. So not, in sort of modern industrial cheese making, they're trying to do a lot to keep microbes out. So, they have a very narrow range of species that they're working with, and they don't want anything else growing. But in the traditional practice of cheese making, you're really cultivating microbial communities from organisms that are growing, like from the environment. So you're not starting with pure sterile cultures. And I think it really speaks to the sort of technological knowledge, again, over thousands of years that cheesemakers developed to understand, okay, well, if we want to ensure these communities grow in the same way every time, we have to really control the environment very precisely for these communities. So, keeping the temperature and the salt and the humidity and everything consistent. And that's enough to ensure this highly reproducible succession of organisms forms on cheeses, day after day.
It makes me wonder, and this could just be, I call it swag for silly, wild guessing, or scientific wild guessing. That would work well too. If you had the original ancestor to some of these cheese-making microbes, I wonder what's changed about them. Have they undergone particular genetic changes so that they're more, and I'm going to use this word in an incorrect way, more social?
Yeah. Yeah, it's interesting. So, I mean, a lot of what we worked on in my lab was trying to understand what are the interactions between species? How do they communicate? Are they fighting? Are they helping each other? Like what's happening? And on a wheel of cheese, there are an enormous number of interactions of different types. One of the things we found over and over again is that microbes are fighting for iron. So iron is sort of a limiting nutrient in many different environments for microbes, but they need it to grow. And it turns out that, it looks like the bacteria were actually like stealing iron from the fungi. They were evolving ways to steal iron from each other. We actually saw another type of interaction that we spend a lot of time working on is the exchange of genetic material between organisms. And I actually have a slide showing a study that we did a number of years ago looking at the genomes of cheese microbes. And we detected, this was done by a former postdoc in the lab, Kevin Bonham, and he detected over 4,000 cases of putative horizontal gene transfer just between cheese bacteria that we had sort of that had been isolated and genomes had been sequenced and put in public databases. And so they're not only interacting with each other to sort of compete over resources and, help each other grow in different ways. They're also evolving on cheese as they're growing together, which I think is really fascinating to think that, these communities, they're dynamic, both in terms of who's growing and, what's even in their genomes.
Despite what I said about this being simpler, this reflects what many people say about an ecological system, right?
Yeah.
So you have a world in a wheel of cheese.
Yes, you do. Yeah.
I'm not a poet. But this idea of evolution taking place while you watch, I describe it as the hand of Darwin is on all that lives, and it's really true, makes me wonder about whether or not a hitchhiker, for lack of a better term, could become incorporated and become part of this collective, this cheese collective. And maybe that's how these things evolve over time. Mm-hmm. But I want to point out that evolution, as you're inferring or perhaps suggesting, takes place within the production of a single wheel of cheese. And that I know that takes place in a culture flask. Why not in a wheel of cheese?
Why not? Yeah. And we actually tried to go a step further and see if we could actually observe horizontal gene transfer during the aging of a cheese. So we had another study that was led by former postdoc Christina Sock. And she, we got samples from Jasper Hill of cheeses aging at like the same, we followed the same batch of cheese over time and got sort of like weekly samples over the course of aging. And she, through a ton of different types of sequencing approaches, including this approach called Hi-C, which is where you try and attach plasmids to chromosomes. So plasmids are these mobile elements that are often exchanged in microbial communities. And we saw a handful of potential examples where a plasmid had moved from one genome to another from the beginning to the end of the time course. I think there's a lot more to be done there, but yes, I think it's very possible that I would say 100% guaranteed that there's evolution happening on these cheeses, but we were able to actually potentially see some of these cases of horizontal gene transfer happening on a wheel of cheese.
And I think it's important for the viewers and listeners to understand that the amounts of horizontal gene transfer taking place in microbial communities is much higher than you might have learned about in intro bio with, transformation, transduction, and conjugation. I'm not saying those things aren't important, but I think it makes it so much more difficult to define taxa, so much more difficult to look at ancestry.
Yeah.
The family tree of a community is much more like kudzu in the sense of all this interaction taking place. And here you're seeing it in a wheel of cheese.
Yeah.
And obviously, people are looking for things like that even in the human microbiome. And
absolutely.
they're seeing it too.
Yes, yes, yeah, and we were inspired by a lot of the work that has happened in the human gut microbiome field, looking at horizontal gene transfer. So yes, it's happening everywhere. It's really fascinating to be able to sort of just observe microbial evolution, almost in real time. And yeah, so it's...
And one of the things that I see from time to time, I'm very interested in, and when I say this, I mean, this is just an interest of mine. I'm no expert. The idea of how microbes interact is very important. And I run into a situation where people more or less infer intentionality, or human ideas to it. So like everything tends toward a positive symbiosis.
Right.
Or every interaction is negative. And when students ask me, is it 1, 2, or 3, I'll say yes. and I would argue that it's no different than anywhere else on the planet. There are positive and negative interactions. It's not that it's all martial or pacifistic.
Yeah.
So you mentioned before that bacteria can use, presumably via siderophores, and I believe that's in one of your slides, are able to steal iron to inhibit the way that fungi grow. Well, the fungi are going to try to evolve to get around that over time.
Yeah.
And then other bacteria might want to make siderophores with a higher affinity for iron. It's a moving target.
It is absolutely a moving target. And yeah, the fascinating thing about what happens with the fungi, and a lot of what we ended up studying in the lab was these fungi because they were doing such cool things. What we saw is that the fungi are really able to manipulate the community, and again, I don't want to assume intentionality here, but they're carrying out very complex interactions that the net result of which end up determining what the microbiome looks like there. So as an example, the fungi they're really good at secreting enzymes to break down macromolecules, large, pieces of food that bacteria may not have access to otherwise. And so they're secreting enzymes like proteases to break down the casein, which is these, large protein complexes. The bacteria can then feed off of some of the peptides that have been released from the breakdown by the fungi. But what the fungi will also do is secrete antimicrobials. And so, that will inhibit the growth of bacteria, potentially protecting themselves from the bacteria eating all of the resources that they're taking the energy, making enzymes and using their energy to break down. But some bacteria have evolved in these communities to be resistant to the antimicrobials. And so if you are a microbe that maybe hasn't evolved in this community, you won't have the resistance to the antimicrobials that the fungi are producing. So you're not going to be too successful. But if you do have the resistance, then maybe you can grow. The fungi are also secreting these siderophores to take up iron. Now, maybe you're in a bacterium in the community and you happen to have evolved the ability to take up fungal siderophores. Now you are doubly, double bonus. So you can both feed off of their leftovers and take their iron. And there's just all of these interactions. And then we started bringing in phage into the community. So Tara Spencer, who was a former grad student in the lab, worked on, okay, what happens if you start introducing phage? What happens to interactions? And we saw this really interesting sort of three-way interaction between the phage and bacteria and fungi, where the phage will want to basically, infect the bacteria to replicate. And then but the bacteria then can evolve resistance by changing their cell surface. But by changing their cell surface, they now become susceptible to the fungal antimicrobials. So you basically have to balance all of these interactions from your neighbors to survive in even just a simple microbial community like this, relatively simple.
And it's interesting to me because this is, if you'll forgive the expression, a microcosm of what we see all around us. Yeah. And it really is, because this community is constantly, seemingly duking it out all the time. But they're also preventing hitchhikers from taking residence in this preferred community. Yeah. You know, it really is that kind of situation where it's two different things taking place at the same time. They're preserving their community and they're also constantly battling.
Yeah.
and that means that they're constantly improving on both sides. And, you know, if you think about it, this is at the heart of the antibiotic crisis, because the antibiotics that we treat disease with, they don't evolve.
Right.
But the microbes that are sensitive, those antimicrobial agents, those microbes do evolve. Mm-hmm. So we see echoes of this everywhere. And it's funny, I don't know if you can estimate the numbers of members in these simple communities. What would you say?
Yeah, it's on the order of, you know, tens or fewer of species. It's, you know, for a Brie, Camembert type community, it's like a handful of species that live together. So,
and isn't that fascinating that they're still able to undergo all this complexity with that? And this is another great example of an emergent property. If you look at them individually, you would not expect this.
Yeah.
And that's wonderful.
Yeah. Yeah. It's fascinating. I think when I got into cheese, I was like, oh, this is a simple community compared to microbial mats, for example. But the more I studied it, the more it became clear that it was going to take a lifetime to really understand what's going on, even in a community that has relatively few number of species. So there's endless things to discover.
How does one go about, or are there no examples of a truly new cheese? Because we've talked about that. Oh gosh, I don't want, you know, evolutionary biologists to get mad at me, but that coevolution that takes place between the members.
Yeah.
How about a new cheese? Have they been coming on the scene, or is that something that only exists after a long period of time?
Yeah, it's interesting. In Europe, there's a lot of protections around these very traditional cheeses, and cheesemakers have to adhere to these very specific ways of making the cheeses and what ingredients they use. In the US, that we don't have those, so cheesemakers have been sort of more free to experiment with how cheese is made, and artisan cheesemakers in the US have created many different variants of these traditional cheeses by tweaking some of the methods and the ways they make the cheeses. So there's actually like pretty big diversity of cheeses available by artisan cheesemakers in the US. You know, there's also hundreds of cheeses traditionally made in Europe. So there's already quite a bit of diversity. Making a new cheese, though, yeah, I think it's interesting. It's an interesting problem because you're trying to, create communities that work well together. And I think in cases where people have tried to, say, introduce a microbe that they think would be good for the flavor of the cheese, it ends up not really being able to colonize because it doesn't have the tools it needs. Yeah, it hasn't co-evolved with the rest of the community. So there's been a couple of studies showing that, you know, these microbes, they just die off. They don't have what it takes to succeed in the community. So you really would have to take a more community-centric engineering approach to do this.
So first, I wanted to say that's why altering the conditions in which you ripen the cheese would cause changes to take place rather than adding things.
Yeah.
But related to that is what you just touched on. Do you think that AI type approaches could be used to create new combinations that would be more likely to establish?
Yeah, that's interesting. I think that's an interesting idea of I think people have thought a little bit about AI or trying to predict like what microbes might be good in terms of flavors that they might introduce. But yeah, could you predict what microbes might live well together? That's a super interesting idea. I don't know. I don't know that people have done that, but I think that's kind of the approach that you would have to take. In one way or another, you'd have to find microbes that live well together, that play well together. And are delicious.
There you go. There you go. So I think this is such an interesting area, and I know that it's kind of transmitted itself to other places and all the rest, but you made kind of a decision the last few years to change your direction a little bit.
Yeah.
And I thought you could take, because we're we've got about 20 minutes. I thought you might want to talk a little bit about that transition.
Yeah. So about 2 years ago, I decided to actually wind down my lab at UCSD and move to a new company called Arcadia. Not as new anymore, but and a lot of that decision had to do with the challenges that I saw really in studying microbes from the environment. The fact that, we could take these communities apart and put them back together, but we didn't have a lot of tools to work with them other than sequencing. We didn't have genetics, which is, what I was trained to do and is such a powerful, elegant way to understand life. So it was hard to really understand what was going on, both from like the development of tools as well as their genomes were just very poorly annotated, I would say. Like, we would try and do all these studies to understand what was happening, and over 50% of the genes that ended up being things that we saw, okay, these are really important for communities, these are really important for interactions, they were genes of unknown function. Mm-hmm. So there's just this big gap in our ability to be able to really truly understand what microbes are doing and what their true potential is for potentially using in other ways, because we haven't really invested in the types of tools that we need to study new microbes that are not the E. coli's of the world. And so a lot of the work that has happened at Arcadia is trying to think about, like, how do we understand organisms more broadly across the tree of life. And so that idea of being able to really focus on this problem of how do we understand diversity more broadly is what drew me to this position at Arcadia. And that has led me to make another move recently to Astera, where I will hopefully be leading my own company to look even deeper into some of these issues involving microbes. And the things I'm especially starting to think about now are fungi, which are things that we spent a lot of time working on in my lab, which have a huge amount of interesting biology, but not a lot of tools to work with, the filamentous fungi in particular, as well as these properties of communities that I think are just really interesting and powerful. So yeah, it's been an exciting past couple of years of me evolving into in different ways.
You're like a shark. You need to keep swimming.
Yeah, I think so. I think so. I really like, starting new things and building things from the ground up. I think that's what I really enjoy doing about building cheese as a system. Doing those first few things of okay, this is an important thing that I think somebody should do. So maybe I'm the person that should just go out there and build it.
Parenthetically, I did my graduate work on what was then an undomesticated bacterium, Cynorhizobium meliloti, and there were no tools really other than some ideas about doing transposon mutagenesis via suicide plasmids, which was very new at the time. I developed a generalized transducing bacteriophage, and that way we could move genes from places to places.
Yeah.
Yes. So when you have a mutant, it's an isogenic one. So the only change is the mutation that you've made. Fungi, and I should say that I've spent most of my career with, quote, undomesticated bacteria, end quote. But fungi are a different can of metaphorical worms.
Yes.
Because they are so unusual and so understudied. When you look around, the idea of someone who's a mycologist who isn't necessarily just interested, and I'm not diminishing this, in medical mycology, which is a big deal,
yes,
but just looking at fungi in general, very complicated. So I tip my hat to you.
Yes, it's they are really challenging to work with. compared to, let's say, E. coli. They grow on solid, they grow as these mycelial structures. There's not a lot of genetic tools for them. There's, pockets of expertise, but not widely distributed, knowledge about how to work with them. But they have amazing biology. They can do these fascinating things like break down complicated molecules. They can form structures. People are using them to do really cool things like actually build structural materials, make textiles, make foods. So there's just a lot of potential, I think, in working with fungi in more detail.
So I believe that you have some interest in potentially some of the actions that fungi have affecting the behavior of other organisms even.
Yes. So I forgot to mention this. When we were at Arcadia, we built up a couple of different microbial research programs, sort of one of which I think ended up being super interesting. So we combined this previous interest in horizontal gene transfer, and they're a fantastic computational biologist, Taylor Ryder, who's at Arcadia, developed a new way to very quickly scan for horizontal gene transfer events. And she then collaborated with Emily Weiss, who coincidentally was one of my first graduate students at UCSD, then a postdoc in my lab, and then came with me to Arcadia and is now a scientist at Arcadia. She is very interested in fungi, especially neuromodulatory fungi. So things like cordyceps, which infect insects and manipulate their behavior. And so we wondered, okay, can we apply this tool to look at horizontal gene transfer to see if there has been HET between any of the fungi that are known to be neuromodulatory and anywhere else? Like, how do they get this ability to produce molecules to have effects on the brains of different organisms? So they work together, and I have this slide showing sort of documenting horizontal gene transfer or analyzing horizontal gene transfer between these neuromodulatory fungi as well as other organisms, and ended up finding this really interesting sort of enrichment of events of transfer between a class of bacteria, actinobacteria, which are known to be really powerful makers of biologically active small molecules, so sort of the professional drug makers of the bacterial world, into these fungi that are known to be neuromodulatory. And so there's just a ton to be done there and learned about, what is the diversity of molecules that these fungi make, how do they do it, and the evolution of this process in general is fascinating.
Here at the University of Puget Sound, after I earned tenure, they told me that now in exchange I had to teach this introductory writing course, and I noticed there wasn't a lot of science being taught. And I mean, no disrespect to the few people who did. And I know that first semester students really need to be made enthusiastic. So I taught a course on symbioses and parasitism, where students picked out things that were interesting to them, and they researched them, wrote term papers, gave presentations, had class discussions. And one of the things that most fascinated folks were the idea of organisms that affected the behavior of their hosts. And there's a particular characteristic called summiting. And the infected organism, usually an insect or of one form, goes to the top of wherever they are. And this is because either one, they're more easily eaten by predators, or two, a good place to spread spores. So cordyceps, for example, is one such organism, but there are lots of other organisms that make some kind of neuromodulating, and I like that term, cocktail, that changes this. So it's apparently something that's happened a lot. Now, I say this long before The Last of Us came out. Okay? Let's be clear. So this idea of looking for fungi that not only seem to affect sociology or social activities within a wheel of cheese, to something that might affect the organisms' behavior is fascinating beyond belief.
Yeah.
And I love this idea so much, and I'm so delighted that you've looked into it.
Yeah, and we hope more people will pick up this work. We've actually published a couple of these pubs out of Arcadia, one on the method of looking for HTT across species that Taylor developed, as well as the results from their Emily and her collaboration looking at these fungi. So lots of work to do there.
And so now you're in the process of thinking about creating your own company, which is exciting.
Yes.
Hence the title of this episode, which is, cheese, microbes, and Brie-ond,
yes.
right? So I'm not going to ask you about what your company is going to do.
Good, because I don't know yet.
But what I am going to do is wish you the greatest luck in it. I hope you come up with an equally cool name.
Thank you.
Rachel, I kind of have to ask, given some of the stuff I showed at the beginning, do you think it's possible to make luminescent cheese?
Yeah, there's actually one of the microbes that occasionally can contaminate cheese. It's not toxic, but it just doesn't taste very good. Is Pseudomonas fluorescens, and one of the ways that cheesemakers will try and see if their cheese has been contaminated by the species is turning the lights out and using a black light to see if there's any fluorescence from the species. In terms of engineering like a GFP type or, luciferase type cheese, sure, why not? We could do it, but I don't know that you want to eat that.
Well, I don't know. I mean, the people who made the firefly petunia that I posted about, that's a subset of genes from a fungus. And presumably you could put that, say, in Penicillium rotatum, and then you would have this glowing fungus. The other thing that I would recommend, put a little bit of a cheese in the luminometer and just see if it's making light on its own.
Right.
I'm curious about that.
Yeah, I haven't done that. So maybe a good project for students.
And I have one kind of final question for you, if I can.
Yes.
What is your favorite cheese and why?
Oh, that's the hardest question, and everybody wants to know. I would say, I love all cheese, but if I had to pick one, for both flavor and sentimental reasons, it would be Bailey Hazen Blue from Jasper Hill Farm. It is, taste-wise, it's creamy, fudgy, salty, yummy, delicious. For people who think they don't like blue cheese, I try that cheese and then tell me if you still don't like blue cheese. For personal reasons, it was our lab rat for a long time. A lot of the early work we did on cheese was from that cheese. The other personal reason is, one of the times I went to go collect samples of that cheese is when I met my husband in the cheese caves. So, so it's that cheese has a lot of special ties for me.
Very interesting. And I wanted to tell you one more thing. Do you read science fiction?
Occasionally.
In the 1950s, I'm going to send you a copy of this book.
Okay.
Frank Herbert, who wrote Dune,
wrote
a novel called The Santaroga Barrier.
Okay.
And it turns out there's this little town where nobody wants to leave, and it's all because of the special cheese that they make there. And what this cheese has done is created a group mind between everyone in town, and they're not aware of it. But the group mind is, so it protects itself. It is a really paranoid read. And the name of that cheese, the name of that cheese is Jasper's.
No.
Yes. I'm going to send you a copy. It's a very old book. May not be your thing, but I'll bet it is.
That's really fun. Thanks.
Oh, you're very welcome. And it has been such a delight chatting with you. And
you too.
I have a number of things that I'm going to write to you about, and you're under no obligation to respond to them, but you've got me all fired up about this stuff.
Thanks so much, Mark.
Okay, I wish you and your family just the very, very best.
Thanks, likewise.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with particularly yummy links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Rachel Dutton is a science resident at the Astera Institute in Berkeley, California. Many, many thanks as always to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.